Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Hello students, welcome to TV learning program. My name is Benon. I will be taking senior 4 students in biology. Students, as you know, in biology, we study living organisms. And these living organisms, some are plants, others are animals. We are able to see some of the organisms with our eyes, but some other organisms, we are not able to see them. We can't see them with our naked eyes. So that's why scientists designed measures of seeing those organisms, because some are good in everyday life, some cause harm to other organisms in everyday life. So it is in that case that scientists developed what we call a microscope. And a microscope is able to make those tiny organisms we can't see with our eyes big, so that we are able to see their structures, we are able to see internal parts of their body. Our lesson today will be from Unit 3, which is microscope, and the lesson will be about compound light microscope. Students, have you ever used a microscope? Well and good if you have ever used one. And if you have never used one, our today's lesson will let you understand different parts of a microscope and their functions. You will also know how you can use the microscope. Before we continue, do we need microscopes in our everyday life? Yes, we do, because we have seen some organisms are small and they can't be seen by our naked eyes. And remember, those microorganisms or small organisms, some are good, others are bad, because they can cause diseases to other organisms. Can we discuss different institutions, different places where microscopes are used? Where do you think microscopes are used? Excellent. Microscopes are used in schools, because in schools we teach biology, where we talk about organisms. And these organisms are made up of small structures which are called cells. Do you understand the meaning of the word cell? A cell is a basic unit of the body. Cells are basic units of the body. And these cells are too small. They are tiny in a way that we can't see them with our naked eyes. That's why we use microscopes to see them, because these cells have different parts which perform different functions. So in schools, if we study organisms that have cells which we can't see with our eyes, then we use microscopes to see them so that we can study them when we are able to see their structures. That means hands-on, mind-on activities. It's not only in schools, even in universities, high institutions of learning, they do a lot of research about different organisms. They also study organisms. So in that case, universities also use microscopes. Hospitals use microscopes because, remember, most of the disease-causing organisms are too small to be seen by our eyes. Here we can talk about bacteria and some viruses. You can't see them with your eyes, so you need a microscope. In hospitals, they use microscopes. In forensic labs or forensic institutions, whereby they can detect crime scenes, they also need microscopes. These forensic institutions are, in most cases, in police. Countries where they have police, they have forensic institutions which are responsible for detecting and discovering people who have committed crimes. So some of the work they do need microscopes. In geology, do you understand what it means when we talk about geology? Geology is the study of structures of the earth. And people who study geology are called geologists. Geologists can study different rocks and minerals which make those rocks. So it is in that case that they may need microscopes. So as we can see, microscopes are very important in everyday life. And there are very many institutions where they are used. We have seen schools, universities, hospitals, forensic labs, and geologists also need them. You can also find or think about other places or institutions which need microscopes. When we talk about microscopes, here we have two microscopes, and I would like to know how they are different. When you see this microscope and this one, you can see that on the upper parts, they are different. This one has only one part, and this, there are two. This one, we call it a monocular microscope. Remember, mono means one, and this one is a binocular microscope. Now, these microscopes have different parts which perform different functions. Let us talk about parts one by one. Here, as you see down, this part, we call it base. And do you know the function of the base of a microscope? The base is used to support upper parts of the microscope and to make it stand stable. We also have what we call light source. When you see here, this one is the light source. Light source. And on the side, we have the switch, which means I can switch it on. I can also switch it off. And when it is on, you can see that this is the source of light. That's why we call it light source. Remember, this one we have called it a compound light microscope, which means it uses light. So the function of the light source is to provide light, which is needed by this microscope. As we go up, we also have what we call stage. We have what we call stage. And the function of the stage is to support the prepared slides. Or in other words, that's where we put what we are going to observe. We put there what we are going to observe. As you go up, we have what we call stage clips. This is the stage clip. You can see it. And the function of the stage clip is to hold the prepared slide, or what we are going to observe. Stage clips. As you can see, we have the stage. We also have stage clips. These stage clips are on top of the stage. Then from there, we have what we call objective lens. You can see here we have 4 of them. They are different in size. These are objective lens. They are different in size. Even the capacity to make the specimen big is different, because on some of them, you see there times 40, or we can say times 100, and so on, which means the capacity to make the prepared specimen big is different. This one can make it 40 times bigger, and this one can make it 100 times bigger. So these are objective lens. And those numbers are written there. If you check nicely, those numbers are there. Like you see here, we have 100, and here we have 40. When we go up, we have what we call eyepiece lens. Eyepiece lens. Eyepiece lens. And you see here, there are 2. That's why we call this one a binocular microscope, because we can use these eyepiece lens to see through them. And these eyepiece lens also have different capacity. Like when you see here, it is times 10. And here it is times 16. Which means the capacity to magnify the specimen is also different. Then on the side, we have what we call coarse focus knob. Coarse focus knob. Coarse focus knob. And the function of the coarse focus knob is to move the stage up and down. You can see this is our stage. It is going up. It can also go down. That is the function of the coarse focus knob. Then, on the side, we also have what we call fine focus knob. Fine focus knob. That one also moves the stage up and down, but slightly, so that the specimen is easily observed through the objective lens. Now that we have finished observing the main parts of our microscope, where we have seen the base, we have seen the light source, we have seen the stage, we have seen the stage clips, we have seen the coarse focus knob, we have seen fine focus knob, we have seen the eyepiece lens. We can start using our microscope. Now, when it comes to using the microscope, sometimes we have what we call prepared slides. That means those are slides which have been prepared by other scientists. We can observe them. Like here, I have a prepared red blood cell. Prepared red blood cell. I can put it on top of the stage of the microscope. And when I put it on top of the stage of the microscope, it is going to be supported by stage clips. The stage clips are going to hold it in position, and then from there, I can observe nicely my specimen. I'm observing the components of a red blood cell. You can see the stage is moving up and down. I'm trying to direct the focus on the stage so that I can observe the red blood cell. This is how I observe my specimen. And sometimes this word is used, but it is not clear. Do you know what the word specimen means? Specimen is sample. The sample to be observed. Specimen is the sample to be observed. Then we also have the muscle cell. This one is another prepared slide, which we can observe using the microscope. We can observe this cell using the microscope. Then from there, these are prepared specimens. We can also prepare our own specimen and then observe it. Here I have the onion. I have onion. I have iodine solution. I have water. And I have gloves. I have what we call slides. You can see on these slides there is no any sample or any prepared specimen. Then I also have these are very small. These are called cover slides. These are called cover slides. Now, I also have a tissue. Then what I do, I cut my specimen. First, remove the outer part. Then I can cut the onion into 4 pieces. Then from there, I can remove one part. Then from this part, I can be able to extract the internal part using the tweezer. After removing the internal part or the membrane, what I do, I get my slide. Then on top of my slide, I put water, few drops of water using a dropper. Then from there, I make sure I put my onion membrane. I make sure it is flat. I make sure it is flat. And the reason why I put there water is to avoid drying and wilting of the cells which make this membrane. Then from there, I have to put iodine solution for staining. I get my dropper. I put few drops of iodine solution. After that, I get my cover slip to put on top of my specimen. I put it on top of the specimen, and I make sure there are no air bubbles. I make sure there are no air bubbles. Then from there, I get the tissue to remove excess water and excess iodine. You can see the tissue is wet, which means there was excess water and excess iodine solution. After that, you wait for a few minutes, and then you put it on the microscope for observation. So we can repeat some of the materials we have used. You need water. You need iodine solution for staining. You need the tweezer. You need the slide. You also need the cover slide to put on top of the specimen. Then you need your onion. Remember, this onion has plant cells, and those are the cells we can see. Then from there, I get our prepared slide, and then I put it on the microscope so that we can see the onion cells. As usual, I put it on top of the stage for observing. Remember, if you put it on top of the stage, it has to be supported by the stage clips. After you have put it there, you can observe your prepared slide. Of course, you have to check, and then you also have to use the coarse focus knob and fine focus knob so that you can see the cells within the onion. Then after that, you are able to identify different parts within the cell. You can see the nucleus, you can see the cytoplasm, you can also see the outer membranes. Dear students, our lesson today was about microscope, and we have discussed parts of the microscope where we have talked about base, light source, stage clips. We have also talked about eyepiece lens, and we have talked about the arm which supports those lenses. We have also seen coarse focus knob and fine focus knob. We have seen that these microscopes are used by different institutions. They can be used in schools, they can be used in universities, they can be used in hospitals, they can be used in forensic labs, and even geologists can use them. Those are not the only institutions which use microscopes. There are also industries and other places which need microscopes. We have seen that in our labs, we can have prepared slides, which were prepared by teachers or other scientists. We can also prepare our own cells. Like you have seen, we have prepared onion cell, getting it from the onion. Then from there, we have homework to test our level of understanding, because you have followed all the steps of the lesson, and then you should be able to answer these questions. This is the homework. The first question says, where are the microscopes used in the society? Where are microscopes used? Are you able to answer that? Of course, if you followed all the steps of the lesson, you are able to answer that. You can give as many places as possible. Question number 2. What are the functions of the following parts of a microscope? Base, arm, coarse focus knob, fine focus knob, then objective lens. Hope you are able to give functions of those parts of a microscope. And then question number 3, explain why microscopes are used in schools and hospitals. Why do schools use microscopes, and why hospitals need microscopes? Students, thank you for your active participation. Have a nice time.